Okay, let's dive into this. Every single one of us is built from an incredibly complex, microscopic instruction manual. I mean, it literally dictates everything from our biological sex right down to the color of our eyes and even how our blood functions. But what happens when there's a tiny typo in that manual? Or, when an entire extra page just gets added in? Welcome to this explainer. Today, we're going to explore the absolutely fascinating world of human genetics, how inheritance works, and ultimately, we're going to focus our lens deeply on the biological mechanics of Down syndrome.
Here's our agenda for today. First, we'll look at the genetic instruction manual and determining biological sex. Second, tracking traits across generations. Third, single-gene Mendelian disorders. And finally, we'll dive deep into chromosomal disorders, specifically Down syndrome. Don't worry, laying this groundwork first is really going to help us understand the deep biology later on.
All right, Section 1, Determining Biological Sex, the Blueprint.
So, before we can really grasp any kind of genetic variation, we absolutely have to know the human baseline. And that baseline, it's 46 chromosomes. You can think of these as the 46 chapters of your biological instruction manual. They're arranged neatly in 23 pairs. Now, the first 22 pairs, so 44 chromosomes in total, are called autosomes. They hold the code for the vast majority of your body's traits. But it's that final 23rd pair, the sex chromosomes, that actually determines biological sex. Now, in humans, and actually fun fact, in fruit flies like Drosophila too, we use an XXX system. Females are what we call homogametic. All that means is their 23rd pair consists of 2 matching X chromosomes. Males, on the other hand, are heterogametic, possessing 1 X and 1 Y chromosome. And because females only produce eggs with an X chromosome, it's actually the father's sperm, which can carry either an X or a Y, that holds the deciding vote on the biological sex of the offspring. If an X-carrying sperm fertilizes the egg, boom, the zygote is XX, a female. If a Y-carrying sperm fertilizes the egg, the zygote is XY, a male. But nature loves variety, right? What is incredibly fascinating is that if we look at birds and reptiles, the entire system is flipped completely on its head. They use a ZZ-ZW system. In their world, the females are the heterogametic ones, meaning they have 2 different chromosomes, Z and W. And the males are the homogametic ones with 2 matching Z chromosomes. No way, right? Let's move on and see how this builds, because nature has some pretty wild blueprints out there. In humans, the real magic of male development happens because of something called the SRY gene, that stands for the sex-determining region on the Y chromosome. When this gene is present, it basically pulls the trigger on the development of male features. If it's absent, the embryo just defaults to developing as a female. But looking outside of mammals, insects have entirely different, almost sci-fi blueprints. Take grasshoppers, for instance. They use an XXX0 system, where males literally have 1 less chromosome. They just have a single X. Or look at honeybees. They use a system called haplodiploidy. Female bees develop from fertilized eggs and have 32 chromosomes. But the male bees, they develop from unfertilized eggs through a process called parthenogenesis and have only 16 chromosomes, meaning they are essentially born without a biological father. Moving right along to Section 2. Tracking traits across generations. Now, what's really interesting about this is how it transforms a standard family tree into, well, a generational mystery map. This is a pedigree chart. Specifically, we're utilizing a flowchart method here to crack the code of inheritance. In these charts, the squares represent males, the circles represent females, and when they're shaded in solid, it means that specific individual is affected by a certain genetic trait or disorder. So by following this logical flowchart, we basically get to play genetic detective. Step 1, we look for vertical transmission. We ask, is the trait showing up in every single successive generation? If the answer is yes, we're likely looking at a dominant trait. Step 2, we look at sex distribution. Are males and females being affected equally? If so, it's probably an autosomal trait, meaning it lives on one of those first 22 pairs of regular chromosomes, not the sex chromosomes. And step 3 is absolutely crucial for ruling out X-linked traits. We check for male-to-male transmission. Think about it. If an affected father passes the trait to his son, it absolutely cannot be X-linked. Why? Because fathers pass their Y chromosome to their sons, never their X. So by answering these questions step by step, we can confidently nail down inheritance patterns, like autosomal dominant inheritance, where you only need a single copy of a mutated gene to show the trait. Which brings us to Section 3, Single Gene Disorders, Mendelian Typos. When we study inheritance, genetic disorders generally fall into 2 main camps. On one side, we have Mendelian disorders. You can think of these as a single tiny misspelled word inside our massive 46-chapter instruction manual.
It's a mutation in just one single gene, and it strictly follows those inheritance laws we just tracked on the pedigree chart. On the flip side, we have chromosomal disorders. Now, this isn't just a misspelled word. This is an entire page or even a whole chapter being completely torn out, duplicated, or glued in upside down. It's the literal absence, excess, or abnormal arrangement of whole chromosomes.
Let's look a little closer at those single gene Mendelian typos first. Sickle cell anemia is a classic example of an autosomal recessive trait. It's caused by just a tiny substitution, literally one amino acid. Glutamic acid is swapped out for valine in the beta-globin chain. This minuscule typo causes red blood cells to become sickle-shaped under low oxygen. Then we have phenylketonuria, or PKU, where the body completely lacks a specific enzyme. This leads to the toxic accumulation of an amino acid that can actually cause mental retardation. But pay close attention to hemophilia and colorblindness here. These are X-linked recessive disorders. Remember our pedigree detective work? If a father is colorblind, can he pass it to his son? Biologically, no. The gene for colorblindness lives on the X chromosome. A son receives his sole X chromosome from his mother and only the Y from his father. So male-to-male transmission of an X-linked trait is just biologically impossible. Females, because they have two X chromosomes, are usually unaffected carriers, which means these particular disorders end up disproportionately affecting males.
All right, Section 4, Chromosomal Disorders, focusing on Down syndrome. This brings us to the real core of our explainer today. We hear a lot about Down syndrome, but stepping away from the symptoms for just a second, we really need to ask, so why does Down syndrome happen biologically? What is the exact physical mechanism occurring deep within the cells? So the crucial point here is that Down syndrome is not a single mutated gene being passed down, like colorblindness. It's actually a chromosomal disorder, technically known in biology as trisomy 21, tri meaning 3 and somy referring to the chromosome itself. Biologically, it is caused by the presence of an extra, entirely whole third copy of chromosome 21. And this brilliantly illustrates exactly what we're talking about. When we look at a karyotype under a microscope, which is basically a visual layout of a person's complete set of chromosomes, we can see this anomaly so clearly. Look at the pairs lined up, 1, 2, 3, all the way down. They're all these neat, tidy pairs of 2. But let your eyes drift down to the 21st position highlighted there. Instead of the standard pair, there is an extra third chromosome just sitting there. That single extra microscopic structure, that is the biological root of Down syndrome. Now, because chromosomes house thousands of genes, having an entire extra one means the body is basically receiving an overabundance of instructions from chromosome 21. And this drastically alters the development of the body and brain. This biological excess results in the specific physical and cognitive features we associate with trisomy 21. Based on our source material, these include varying levels of intellectual disability, a flat facial profile, a small round head, a furrowed tongue, a shorter physical stature, and a single palmar crease straight across the hand. Every single one of these traits cascades biologically from that one extra copy of chromosome 21 we just saw in the karyotype. To fully grasp the sheer impact of these whole chromosome variations, it really helps to compare Down syndrome to other conditions. While trisomy 21 affects an autosome, we also see variations in the sex chromosomes. For example, Klinefelter syndrome occurs in males who inherit an extra X chromosome, resulting in a 47XXY karyotype. This extra genetic material leads to the development of feminine characteristics and sterility. Conversely, Turner syndrome occurs in females who are completely missing one X chromosome, what's known as a 45XO karyotype. This absence of genetic material results in stunted growth, underdeveloped ovaries, and sterility. So whether it's an extra autosome like in Down syndrome or an extra or missing sex chromosome, the addition or subtraction of these massive genetic chapters drastically rewrites a person's entire biological outcome. You know, from a single substitution of an amino acid in sickle cell anemia, all the way to the addition of an entire extra 21st chromosome in Down syndrome, our biological reality is just incredibly delicate. We are all bound by these microscopic blueprints, and yet no two of us are exactly alike. Which really leaves us with a profound question to end on. How does the addition or substitution of just microscopic cellular material, stuff that is completely invisible to the naked eye, shape the vast, beautiful, and complex diversity of all human life? It's a pretty amazing reminder that inside every single one of our cells, there's a whole universe of information just waiting to be explored. Thank you so much for joining me for this explainer, and keep questioning the microscopic world within you.